So, how are you, students? How are my dears? So today we'll start with the experiment to show the effect of the substrate on the enzyme activity, as I explained in the theory. So there is a maximum activity, which is called Vmax, as I explained in the previous video. Here we have, if you add more substrate, you will get the Vmax after certain amount of the concentration. Of course, we can calculate the Km here, which is half of the Vmax. All this explained in the theory part. So today we would like to prove this graph here in the test, how to effect of the substrate reagent upon the rate of an enzyme reaction by using the Mechanism-Menten equation. So our procedure today will start with preparing 9 tube, and we will prepare the concept of the test as I explained in the theory part, that we have a protein. When we add a pepsin in acidic media, we will get a polypeptide. This polypeptide can be reacted in the basic media with fulin conchesto, will get a purple color. So if you add more concentration of protein, even with the same amount of the enzyme, what will happen? Up to a certain part, there is no more results. It will be get the maximum activity, which is called Vmax. We need to prove this one here. So we'll start with the preparing 9 tube, as mentioned here on the table. I need everybody to please open your manual. So here we have, open your manual with this video, please. We will have this tube, 9 tube. We will prepare this 9 tube. Number one, we will add the protein in a different concentration, like 0, 0.5 ml, 0.75, 1 ml, 1.5, 2.5, 3 and 24, 3.25, 4 plus 4 ml plus 5 ml. This is only the protein, and I will add the acidic media, as explained in the theory of the lab. We will add acidic media, which is the HCl, in parallel. So here, after you add the protein and HCl, you need to calculate the concentration of hemoglobin, as I explained before. How come to use CV, C dash, V dash? This is the same concept that we explained before. Don't forget here, we have CV equal to C dash, V dash. I explained it again in the theory part. After that, we will mix the tube. After we mix the tube, we will prepare another 9 tube with a filter funnel tube. So let us start the practical here. We are preparing, this is our setting. We have a 9 tube. In this part, we have a 9 tube, and we have another 9 tube and third 9 for if we need them. So number one, we will mark them as we put here. So this will be the required for the test. That's we need all this one. We need 7% TCA, we need HCl, NaOH, protein, which is hemoglobin today, and the pepsin plus fullerene reagents. Let us start step by step. This is the step number one. We will start by adding the protein as per this table here, the table I mentioned. So this is the table I mentioned. So we need to add 0, 0.5, 0.4, the protein. So here, I add the protein. Here in this part, we will add the protein here, 0, 0.5, 0.7.5. As mentioned here, the amount of the protein and the amount of protein become increased, become dramatically increased until we have 4 and 5 ml of the protein. This is which is here, the substrate. So look here, we mark the tube 1, 2, 3, 4, 5, 6 here, 7, 8, to avoid any misconception for the sample. Here, we finish the tube, we prepare the protein. After that, we will add the HCl here. As mentioned also, please open your manual with me. As mentioned here, we will have HCl. Again, we will add the amount of the HCl by the same amount here in the table, in the parallel tubes. So tube number 1, tube number 2, 3, 4, and so on. So back here to the tube, we add already the HCl for the tube as per the table. So now what I have here, I have, look to the color for the HCl and the protein. So now I have the protein plus HCl. So this is the ready media if you add L-pepsin. That's why the following step here would be the third step. I will add L-pepsin. Here, this step is very important. Very important. Why? When you add a pepsin to each tube, I need to start timing, because I need to calculate only 10 minutes exactly. Is it clear, guys? So here, we need to start with adding pepsin, with 1 ml of pepsin for all of the tube. This is the pepsin here. We will add from this pepsin 1 ml, from this pepsin 1 ml of this pepsin for each tube. Now the reaction will start. So here, back. So now we will start adding the pepsin. Here I have the tabs. Here it's arranged for 1 ml. For 1 ml, now I will start with adding the pepsin. So When I start adding the pepsin, please put your timer and start on. Okay? So now I start step number 1. Now I prepare the timer. Now I will prepare the timer 1 second. So I'll prepare the timer here. Now look to the timer here. I prepare the timer on. Start on. So first 1 ml. So again 2, 3. It's okay, guys. 4. For all of them 5, 6, 7, 8, 9. 7, 8, 9. Here, when you add the pepsin, I already start the timing. So I will wait till the timer will become 10 minutes. And this is very important. We have to mix. So I will throw, this one is here. I will mix. I have the mixer. I will have it. I will keep it here for 10 minutes. What will happen during the 10-minute incubation? There is a digestion happen for the pepsin. Pepsin will digest the protein in the acidic media. As we understand, the pepsin, the only protein which work in the pH 2, as you understand all of this information. So here now, I explained this in the theory part. Here now, I was waiting for 10 minutes. Now, guys, after 10 minutes, what I will have this solution with me now. This is after 10 minutes. After 10 minutes, I will go to the following step. Again, please refer to your manual. You will find in the manual that we are adding TCA. TCA will be trichloroacetic acid. This is 7% trichloroacetic acid. The trichloroacetic acid actually has 2 functions. Number 1, 2 functions. The trichloroacetic acid, and usually we get a question in this part. Number 1 is to stop the reaction. Number 2 is precipitate the protein. So we have 2 functions for the trichloroacetic acid. Now we need to stop the reaction. We need to see what happened, how the enzyme activity work on the substrate concentration, different substrate concentration. Now we add 8 ml of the trichloroacetic acid. So again, I will not add here 1 or 2. We add more 8 ml. 8 ml. Again, refer to your manual. So again, add 8 ml of the trichloroacetic acid. What will happen here? You will find the solution became turbid. That means the protein is precipitated. So it's fine. Now I will go to the following step as per the procedure in the manual, which is called filtration. So I will filter each tube. It's tube number 1 in a tube number 1. Tube number 2 in a tube number 2. Tube number 3 in a tube number 3. Tube number 4 in a tube number 4. And I will make the filtration. So after you finish the filtration, you will get the filtration here. So they will get your filtration here. After you get the filtration, you will just take only 5 ml from the filtration. Just 5 ml. We will put a marker for you. Here we have a marker. That means up to filtrated. Up to this marker, that means this is 5 ml. So when it gets 5 ml, put it in the other rack. So I will have again 9 tubes with the filtration. In this step, what they have, I will have polypeptide because I add protein, acidic media with pepsin. That means I will get polypeptide. I precipitate all the remaining protein. So the remaining in the supernatant will be, ya shabab, will be polypeptide. So the filtration here will contain your polypeptide. I will go to the second step of the equation, which is if you add a fullin to the polypeptide, you will get, which is called purple color. Perfect. So, but this need to be in basic media, I mean NOH. So, now the filtration, first of all, I will add NOH. Again, guys, if you look to the manual, understand what I'm saying. So here I will add 5 ml of NOH. I will add 8 ml, sorry, I will add ml for the NOH. I will add, here what I have in this tube, yalla tell me, what I will have in this tube? I will have 5 ml of filtrate plus 8 ml of NOH. Is it clear, guys? Now I'm doing the media. So here in the second tube, I will have 5 ml from the second tube filtrate plus 8 ml of NOH. The same here, the tube number 6. I will have 5 ml from the tube number 6 filtrate plus 8 ml from the NOH. So I will put the media. After that, I will add the last step 2. Here, this is now ready for the Folin congestion reagent. Here, this will be the last step. I will add Folin reagent. Is it clear? Perfect. So, when you add to the Folin reagent, look to the shape for the colors. look يا بنات. So the color become start increased, gradually increased till here will be stopped. That means here we reach V max. So here now we have a different concentration of substrate. It increases enzyme activity until here, but here it became يا شباب V max means this is the maximum activity of our enzyme. So we need to prove this practically. I will measure this one here through the cuvette. I will take the first tube with the cuvette and I will put them together. I will put this them like here, and I will open the spectrophotometer, and I will put it in the spectrophotometer, and I will take my reading. Here, look guys, to the reading that we have. Here, this is the reading, that's what we get from the result. Look here, we have all these readings. So start the Vmax starting from the tube number, يا شباب, tube number 6. So here, again guys, now يبقى back, now we prove يبقى here. So we remove it from the spectrophotometer. I will make a small brief. As usual, we start with the setting with the tube. We start at the setting on the tube. We add the protein as per the table. We add the HCl for doing the media. After that, we add 1 ml of pepsin, and we accurately put the timing to make 10 minutes. Here, there is a digestion will happen. The protein will be converted to polypeptide. After that, I need to stop the reaction. So I will add trichloroacetic acid with 8 ml, so the reaction stop and the protein precipitated. So I need to remove the extra protein. I will make a filtration. After the filtration, I will have 5 ml from each filtrate, from each tube, gradually from 1 till 9. At the same time, I will add 8 ml of NaOH here. So I will have here 5 ml of filtrate plus 8 ml of the NaOH. So I did the media. After that, last step, I will add the full reagent, which is getting us the color with the polypeptide. So the color gradually increase until the last 3 tubes become fixed, which is called V max. Thank you guys. Inshallah, I will share this video with you on the Blackboard. If not, I will share it with YouTube. Thank you guys. See you inshallah next lab after we come back. Thank you. Bye.